Welcome to an overview of current good manufacturing practice, or CGMP, for sterile drug products. We will focus on the FDA's 2004 Guidance for Industry, which provides the regulatory framework for aseptic processing. The primary goal is to ensure the safety and quality of products where terminal sterilization is not feasible. The FDA distinguishes between terminal sterilization and aseptic processing. Terminal sterilization is preferred because the kill step occurs after the product is sealed. In contrast, aseptic processing is a high-risk process. Components are sterilized separately and assembled in a controlled environment with no final sterilization, meaning any CGMP failure can pose a life-threatening risk to patients. The critical area is where sterilized products in containers are exposed. This zone must maintain a limit of no more than 3,520 particles per cubic meter for particles 0.5 microns or larger. This is achieved using unidirectional laminar airflow at 90 feet per minute, which effectively sweeps particles away from the filling area. Facility design is a fortress, using positive pressure cascades to keep contaminants out. The FDA requires a minimum differential of 10 to 15 pascals between different room classes, ensuring air always flows from the cleanest areas toward the less clean ones. Personnel are the greatest variable and the largest source of contamination. Movement must be slow and deliberate to avoid air turbulence. Proper technique is vital. Never reach over open product. Full gowning is required, ensuring no hair or skin is exposed. We must also control invisible contaminants like pyrogens. Glass undergoes depyrogenation using high-temperature dry heat, targeting a 3-log reduction in endotoxins. Rubber closures are prepared through multiple water for injection wash cycles. Minimizing the time between washing and sterilization is critical to avoid microbial growth from residual moisture. For liquid products, we rely on sterilizing grade filters with a 0.2 micron pore size. These are validated using the challenge organism Brevundimonas diminuta to test the filter's limits. Validation requires demonstrating that a high concentration of organisms results in a completely sterile effluent, meaning no passage. To prove our process works, we conduct a media fill. The drug is replaced with a growth medium, simulating worst-case conditions, including maximum personnel and long durations. Initial qualification requires 3 consecutive successful runs, followed by routine maintenance every 6 months. The goal of a media fill is 0 contamination. For runs under 5,000 units, 1 contaminated unit requires revalidation. For larger runs, 2 contaminated units require full revalidation. Regardless of these numbers, recurring contamination is an adverse trend signaling a loss of process control. Equipment is sterilized using moist heat or dry heat. Validation includes mapping the chamber to find slow-to-heat locations. Biological indicators, placed in the most difficult to sterilize spots, prove a sterility assurance level of 1 in a million. The environmental monitoring system is our radar. We monitor active and passive air, surfaces, and personnel glove prints. This data helps us establish alert and action levels, so we can identify potential contamination routes before they affect the product. A critical lesson is that you cannot, test quality into a product. Statistically, a standard sterility test will pass 98% of the time, even with 0.1% contamination. Therefore, a positive sterility test is a major failure and can only be invalidated if the lab is proven to be the source of the error. Before any batch is released, the quality control unit must review all records. This includes environmental monitoring data, sterilization charts, filter integrity tests, and intervention logs. This final review assures no errors occurred and that all specifications were met. In summary, sterility assurance is built on 4 pillars: sound facility design, controlled inputs, validated processes, and rigorous monitoring. Sterility is not a test result. It is the outcome of strict, unwavering adherence to validated processes